Hi guys, so this is Christina, and I'll be telling you guys about natural killer 92 cells today. I'll be calling them NK92 and NK cells from now. They're the same thing. So naturally, your immune system has NK cells, which act in your first line of defense in the immune system. Natural killer cells are part of the innate immune system, meaning that they aren't specific to their target. Okay, so let's get started. Before I tell you about NK92 cells, I'm going to be going over some important immunology topics first. So, first of all, your natural killer cells are cytotoxic. They have small little granules drawn in yellow that contain proteins such as perforin and also have proteases, which are enzymes that function in the digestion of proteins known as granzymes. And I drew these in red. So when a natural killer cell approaches a cell that either contains viruses or is recognized as a tumor, the NK cell will release its perforin inside of its granules, which will then penetrate the target cell. So what I'm drawing right now is the breaking of the granules, and this would be the release of the perforins that would kill the target cell. Okay, so to draw that a little bigger for you guys, so that is the target cell, and the red dots represent the perforin that split open the cell membrane of the target cell. In this case, it's a tumor cell or a viral cell, and this will lead to the death of it. And the killing of target cells using this method induces apoptosis, which is cell-mediated death. This is way more favorable than necrosis, which is the unnatural death of cells, and this can have detrimental effects on the cells around them, often killing healthy cells. Okay, so another important question to ask would be, how is apoptosis induced? Okay, so that's going to be our target cell. On the target cell are these tiny orange rectangles and with yellow ovals on top of them, which allow the NK cell to recognize the target cell. So when a cell is abnormal or virally infected, the abnormal host cell will present abnormal antigens on its cell membrane, which will be recognized by the immune system or natural killer cells. Basically, the tumor is waving a bright yellow flag to the immune system, which allows the immune system to recognize it and attack it. The yellow ovals represent a protein or antigen, and the orange rectangle represents the major histocompatibility complex, which presents the antigen. Okay, so something important to note is that high levels of MHC protein inhibit NK cell activity. So NK cells are only able to attack the target cells that have low levels of MHC protein. This is bad because it's a loophole for cancer cells to escape the NK cells, and also cancer cells use immunoediting to escape and prevent the attack from NK cells, which allows them to grow and become deadly, but we will not be talking about immunoediting. So back to the picture of the cancer viral cell here. So here's a natural killer cell. They have receptors that attach to antibodies that locate the cancer or viral cells. Those are the receptors. And in red, I'll draw the antibodies. First, the antibody will bind to the antigen that the MHC is presenting on the cancer cell. Then, through the receptors on the NK cell, the cell will be drawn to the target cell, and the NK cell will then induce apoptosis on the cancer cell. And that's how apoptosis is induced. And just to mention, the receptors on NK cells have two different functions. First is to put the NK cell to sleep, making sure that the NK cell doesn't go attacking everything that is in its path, and second is to put the NK cell to work, making sure that the NK cell attacks the appropriate cells and does its job. Yay, so we now know the basics of immunology. Now I'm going to tell you guys about the new research behind natural killer cells called NK92 cells. First is the ANK cell, then is the HANK cell, and last it's the TANK cell. So scientists found a way to manipulate the natural killer cell in order to meet the needs of people with a severe form of cancer or some other viral disease. In other words, these manufactured natural killer cells are being used to fight cancer and viral diseases. So, first of all, let's go over ANK cells. ANK stands for Activated Natural Killer Cells. Basically, the ANK are natural killer cells that lack the ability to turn itself off, meaning that it attacks anything and everything in its path. So you can think of ANK cells as a template for the other two types of natural killer 92 cells, HANK and TANK. We'll first go over HANK cells, also known as high affinity natural killer cells. These HANK cells have a cluster of differentiation molecule on their surface, also called CD16 receptors. So, remember how I said that NK cells are only able to attack host cells that have low levels of affinity? Well, these CD16 receptors allow the NK cell to attack cells with very high levels of affinity, hence, the name of the cell is high affinity natural killer cell. And this is really useful because only 1 out of 10 people are born naturally with these CD16 receptors. These HANK cells will provide the other 9 the means to actively kill tumor and viral cells. The HANK cells are also equipped with IL-2 receptors, which induce quicker killing of the tumorous or viral cell. And I drew these in purple. The second type of cell that was derived from the ANK cells was the TANK cell, which stands for Target Activated Natural Killer Cells. As their name states, TANK cells are target specific and can selectively recognize and kill abnormal or harmful cells. They have a chimeric antigen receptor, also known as CARs, to allow them to function this way. This enhances the function of the natural killer cells since normally they are part of the innate immune system, which only consists of defense mechanisms that are nonspecific. So in pink here, I'll draw you guys a tank cell, and the green squiggly lines are going to be our CAR receptors. And as I said before, CAR receptors are target specific, and normally an NK cell is not target specific, which is the new feature added on. And yeah, this is what allows the NK cell to recognize the specific targets. Okay, so there are the 3 types of NK92 cells that scientists have created to enhance the body's immune system. So, NK92 cell research is fairly new and it's still ongoing today. Most of the articles that I've read and the research papers that I read were from about 5 years ago or even less. So NK92 cells are a part of NantKWest's Cancer Breakthrough 2020 campaign, which is an ambitious and progressive campaign that sets the goal of curing cancer. And through this program, pharmaceutical companies, research institutes, major cancer centers, and also oncologists from all over the world are able to come together to share their data and collaborate in order to ultimately cure cancer. So the collaboration leads to more information for all researchers and more opportunities for patients to participate in clinical trials in hopes of finding the cure for their illnesses. So far, human clinical trials have been conducted using these newly developed NK92 cells on renal cell and melanoma patients by Dr. Hans Kinglemon and Dr. Sally Arai at the Rush University. However, I think they only reached about phase 1 or phase 2, around there. So we still have a long way to go. And their data from these trials show very mild fever reactions from some patients during the clinical trial, but overall, the NK92 cells were very well tolerated by the patients. And even patients that were in advanced stages of cancer saw clinically significant responses to the treatment. And also at the Pittsburgh Cancer Center, a study enrolled patients who had acute myeloid leukemia, and these patients were at high risk for tumor lysis syndrome, where large amounts of tumor cells are destroyed, being released into the bloodstream. But thankfully, no signs of this was shown during the clinical trial with NK92 cells, and some patients even saw a decrease in their blast count, which is really favorable since high blast count indicates some abnormality in the body and indicates that your body is fighting off some infection. So overall, NK92 cells have seen very positive results in clinical trials, and there's a lot of hope for them. I personally believe that the future of NK92 cells is pretty bright, and we should have a positive outlook on them due to the clinical trials that have proven them worthy of further research and speculation. Also, they have been proven to show less side effects than the traditional treatments for cancer, such as chemotherapy and T cell therapy, and they have shown to be very efficient in targeting cancer as well. So, NK cells are a very good method of treatment because, first of all, there's less risk of cytokine storms, which is when cytokines and white blood cell levels are dangerously high. And second, clinical trials have shown less side effects and more success as of now. Some improvements that the NK92 cells are going through is scientists are currently making them useful in combination therapy, which includes other drugs, to show synergistic effects that NK92 cells and maybe some cancer drugs have together. And NK92 cells are also currently an off the shelf treatment, which means that they're not tailored to each individual patient, and that is being improved upon by development of a new type of NK92 cell called HIPSE. And like I said before, NK cells have been shown to be very effective in the treatment of cancer, but still they have a really long way to go before they become a global treatment that everyone has access to. Since it's very new research and has not passed the clinical trials yet, the costs and accessibility to all people is hard to say, but hopefully in the near future we will be able to see many advancements in the NK92 cells. And that's it, guys. Thanks so much for watching.